What was that all about? That one is an ancient chant, and it acknowledges the uncountable manifestations of akua from the microcosm within our na'au that guides our decisions to the uncountable manifestations of living elements that make up the universe around them. It calls to all of them and says, "Eia kahuho ike nui la." Here is the great knowledge that is conferred upon us, and that is what we're here for tonight.
Mahalo-san. Aloha. And welcome. I'm Michael Bruno, Provost here at UH Manoa. And we want to welcome you to a very special event. This event is in part sponsored by the Better Tomorrow Speaker Series, one that, as the website describes, offers incisive talks on the most important questions of our time. Like tonight, I can't think of a better speaker living up to that grand challenge than Dr. Lita Proctor, who will speak with us shortly. I'm here really primarily to say thank you, and I have to begin by thanking our own Professor Robert Perkinson, who has boundless enthusiasm, I always say contagious enthusiasm and dedication to his university, and thank you, Robert, for arranging this. So I want to begin by thanking our primary partners. They include PBRC, which for those of you, I know there are folks here who are not from the university, so that is the Pacific Biosciences Research Center, as well as Kamehameha Schools and the Hawaii Community Foundation. Those are our primary sponsors for tonight. We also have additional sponsors, SOEST, short for the School of Ocean and Earth Science and Technology, Kapiolani Medical Center for Women and Children, the John A. Burns School of Medicine, the Bishop Museum, CTAR, short for the College of Tropical Agriculture and Human Resources, and last but not least, CMIC, which stands for the Center for Microbiome Analysis through Island Knowledge and Investigation. But you can go to their website and practice that one. I do want to say, well, you know, I'm here to introduce Dr. Margaret McFall-Ngai, who will then introduce our speaker. And I told Margaret, well, I could read your bio, which is on a different sheet of paper, or I could tell them a little bit about CMIC, and she gave me the answer. I knew she would. Don't talk about me, talk about CMIC. I know that a number of our faculty who are involved in this center are here tonight. It's really an extraordinary group that builds on the extraordinary strength that this university has in particularly the environmental microbiome. A lot of it centered on the ocean, but now growing and to extend up into the land and well up into the ridge. So we call it ridge to reef, and some, like our own Dave Carl, remind us it's ridge to ridge, because we can go way off to the deep ocean. So the Simaiki, and I'm going to describe this from the website, includes dozens of faculty from across 5, and I think now 6 different colleges across campus. And they are, as the website describes, fostering cutting-edge, transformative, and innovative pono-biological research through the understanding of holistic systems, integrating many forms of knowledge across dimensions of people, culture, history, and place, as it relates to the complex microbial dynamics of ecological systems. So I can translate that to say, this is a uniquely Hawaiian approach that seeks to learn from this place in order to change the world. And I don't think that's an overstatement for what this group is poised to accomplish. So it's now my privilege to introduce Margaret McFall-Ngai. She is professor and director of the Pacific Biosciences Research Center here at Manoa, and she is the visionary behind the creation of Simaiki. Margaret.
Oh, thank you, Michael. That was really a nice introduction. I really appreciate it. And I'd like to thank all my colleagues in Simaiki and for coming and getting together and beginning to think about the things that Michael mentioned. It's my great pleasure to introduce Lita Proctor for this talk, and I'm going to give a little bit of a detailed biography of Lita, because I want you to know this person, because she's amazing. She's a Renaissance person, in my opinion. So before I get started, though, I would like to thank the people who helped arrange her visit, and particularly Robert. So I just wanted to say that again also. So I should mention that in addition to the lecture that she's giving here tonight, Lita is giving talks at Kapiolani Women's and Children's, John A. Burns School of Medicine, Kamehameha Schools, Bishop Museum. So she's been extremely generous to Hawaii, in my opinion, on her visit. Okay, so then who is Lita Proctor? I think you'll be amazed. Like I said, she's a true Renaissance person. So starting at the beginning, she was born in Erie, Pennsylvania. Her mother, if you can see, she's Hapa. And so her mother was an exchange student that came to Erie, Pennsylvania from Japan, not speaking a word of English. And she went to a girls' school there and then began to understand some English because she met an English Irishman from a boys' school. They got married and had children, and one of them was Lita. Lita had a rich childhood. She lived all over the world, including Cambodia, Vietnam, Japan, and the U.S. And at the end of her teens, she graduated from Friendly High School in Washington, D.C. Lita attended Virginia Tech, Virginia Polytechnic Institute, as an undergrad. There at VPI, she did her very first research. She studied the ecology of dry valley lakes in Antarctica, the only ice-free regions on that continent, and she was there for 2 full seasons doing that. Then after she graduated and worked on, after working for the EPA for a while, she went to the University of Georgia. And at University of Georgia, with Professor Larry Pomeroy, she worked in the famous Odum School of Ecology. And her work focused on transitions for those of you who don't understand biology, I'm just going to speak to the biologist for a second. She focused on transitions of microbial metabolism in stratified lakes. And there she met her partner, her life partner, Emily Carter, who's a forest ecologist. She went on to get her PhD at Stony Brook with Jed Furman. And the name of her thesis, her PhD thesis was, or the title was, The Role of Bacteriophages in the Ocean's Microbial Food Web. Now, for those of you who aren't biologists, a bacteriophage is a virus that attacks bacteria. So it's microbe on microbe. So it's very small things. For this work, what she did was she was the very first person. I mean, this is remarkable. She was the very first person to study phages in open ocean systems. She was the first person to quantify phage populations in the ocean and the first person to determine the proportion of bacteria that are infected by phage in the ocean. And so for this work, she published a benchmark paper in Nature magazine, the premier journal of science, for those of you who are not in science. She postdoced with Rob Gonzalez at UCLA to cross-train in molecular biology. So she got her ecology creds, and then she went over to get a little bit more training in molecular biology. Then after that, she took a faculty position in the Department of Oceanography at the Florida State University and was promoted in the year 2000, but then she was sirened away by the NSF to the National Science Foundation to Washington, D.C. to be a program officer in geosciences there. And she was she was a program officer in the Biological Oceanography Directorate. Then she was asked by the Moore Foundation, which was just getting underway, to come on over and help form the Moore Foundation as a philanthropic association for science, which she did for a few years. Finally, she was asked around 2008 to come and head up the Human Microbiome Project at the National Institutes of Health. Well, Lita's an oceanographer. At first she said, no, I'm not doing that because this is not my thing. And so they said, well, okay. And then they came back a year later and said, please, please. And so at that point she said, yes, okay, I'll come on over. And she was persuaded by their arguments to help administer the $215 million Human Microbiome Project. So Lita has come here today to talk story about this topic. And so I'd please like you to join me to welcome Lita to the University of Hawaii.
Thank you. I was starting to cry, so I just need to collect myself a little bit. Thank you, Margaret. So I mentioned out here that my password is bug lover. Don't try to hack into my bank accounts. There's not much money in it. But it really conveys absolutely my passion for microbes. I was serious when I said when I was an undergraduate and I saw those swimming things under the microscope, I was absolutely flabbergasted. So that moment that happened has just marked my whole life. I may just cry through this talk. And so what I really want to do for you today, because I want you to really feel the beauty of microbes, is sort of go through that. Now, I am a bureaucrat, a wonk, so I will show data, okay? I will show government data, so forgive me. But part of the reason I want to show government data is to wow you about the amount of money that has already gone into this field. And frankly, it's just going to be the tip of the iceberg. So please forgive me in advance if I show you graphs with dollar amounts. So I decided to call this Listening to the Microbes song because I was coming to Hawaii and trying to make some kind of connection with the Hawaii, so I think about whale song and all that. But it also describes how we're doing this work. We're trying to delve into these communities, peek into these communities, listen to what they're doing and who they're talking to. So I hope that Listening to the Microbes song is still evocative of what I want to try to convey. So I'm going to start with first principles. What are microbes? And the reason I ask is it's a sloppy term. It's used in a lot of different contexts. It's okay to use it, but understand it doesn't just mean bacteria. It means bacteria, it means viruses, it means all these pictures I have on the wall here. And really, it's microscopic life. And the beauty of microscopic life in nature is they don't live alone. So they don't grow well on petri dishes because they don't live alone. They require microbial partners. And when they work as communities in partnership with other microbes, they interact in communities, they do wonders. They get together and they can break down, metabolize on anything. So that's the beauty of microbes in nature. And so they look more like this. Okay, a big gamish of different species talking to each other, sometimes in intimate contact, sometimes far away, but always the talking to each other and trying to figure out a way to metabolize a substrate. Okay, so when you isolate them separately, you remove their partners. And that's probably been one of the most important things that we've learned about the study of microbes is we deceived ourselves as to what they're doing because we removed all the partners in that conversation. So much of modern microbiome science is trying to keep them together and trying to understand how they are conversing, how they are singing, if you will, together. And in fact, we live in a microbial world. This entire planet and all the life forms on it rely on microbes. Okay, Earth formed 4.6 billion years ago, but we have evidence of life on this planet, all microbial, arising at about 3.8 billion years ago. And in fact, over half of our planet's history was dominated by only microbial life forms, about 2.3 billion years. Wait, I want to go back for a second. And microbes did all kinds of things, but maybe one of the most fundamental things that microbes did in these early period when they were conditioning Earth is they produce oxygen. Photosynthetic microbes produce oxygen, okay? And once oxygen was produced, that really allowed for the development of much more complex life forms and multicellular life forms. So then you start to have all these different multicellular life forms arising again from a microbial function, microbial symbiosis. You already know that nuclei, mitochondria, and chloroplasts and so on are as a result of microbial symbiosis back in the day. And most of the genetic diversity in the tree of life is microbial. So many of you probably see the old Haeckel tree, which looks like a literal tree with man on top, but is a modern way of looking at life on this planet using genetic tools and evolutionary relationships. And we're down here in the corner, okay, in a tippy corner. And even much of this eukaryotic arm of the tree of life is microbial, and all that stuff at the top is microbial. We don't see it. That's part of our blinders, right? We don't see it, but this Earth is dominated by microbes. And of course, microbes live in symbiosis with all life. We either rely on microbes or they live in intimate contact with plants, animals, and humans. But of course, what's happened through human history is that we didn't know about the good microbes because most of our major contact with microbes has been the bad ones. Right? If you're just a casual reading of human history, there have been many, many plagues and epidemics that have done major damage to entire societies, entire civilizations. So we kind of, over decades and hundreds and thousands of years, we kind of got the idea, oh, no, these things are bad, they're all germs. And I just named a few epidemics and so on. But with people who developed vaccines or antibiotics, we started to tackle these infectious agents, these pathogens. But our focus was still on thinking, okay, these things are all deadly, all pathogens. But starting in about the 1970s, I think most of the early work was actually in the water treatment plants and places like that, practical reasons, trying to figure out whether in fact the treatments actually did clean water. There were a number of important direct studies of microbes in nature. Rather than trying to grow them on a petri dish, go right to nature and see where the microbes are. So in the 70s, we started a whole suite of approaches to directly study microbes in nature using stains and dyes and so on. And then as a result of a real revolution that was happening in another field around the evolutionary relationships of life on the planet, we were able, in starting in the 1980s, build probes that were specific for particular groups of organisms and begin to study bacteria in nature using what's called, in the most classic one, is the 16S ribosomal RNA analysis of bacteria in nature. You actually develop fluorescent dyes specifically for bacteria, known classes of bacteria, and then you take those dyes and you look in natural samples and you can count them. Then starting in the 2000s, there's always a lag. All the ecologists and environmental scientists get the techniques out first, and then somewhere along the road come the biomedical types who adopt the techniques for studying the human. So in the 2000s, you start to see that same kind of analysis happening in the human. And so that's when we started to say, oh, look at this. There are now a huge plethora, an entire garden of microbes growing all over the human body. And in fact, in 2007, the National Academy wrote a report to really laud this and to really argue that this discovery of this diversity and richness of microbes in nature that are not germs, that are not pathogens, are almost unknown to us, but now we have the tools to begin to study them. So let's do a little bit of comparison and contrast here for a moment. Most of us usually think of microbes as germs, but actually if you count up all human pathogens, could be viruses, could be fungi, could be bacteria, there's only a little over 1,000, I mean, you think about all the impact that these germs have had on human society, it's a lot, but there aren't that many of them. That's my point. And that's actually against a background of trillions of microbial species all over the place, okay? So this little tiny minority has done a lot of damage, but it's also caused us to not pay attention to the others that are the beneficials. Okay, so my point is that in fact the majority of microbes don't cause disease. In fact, many are beneficial. And microbes on Earth do all kinds of things, okay? They produce soil and regenerate soil. Yes, physical weathering can produce soil, but without microbes, soils can't be nutrient rich. It's the microbial activity in soils that makes soils rich for crops and plants. Oxygen production. Let's take 2 breaths. That second breath came from the ocean, from oceanic microbes, photosynthetic microbes producing oxygen. So 50%, approximately 50% of the oxygen on this planet is produced by these invisible to your eye, but hugely abundant microbes that live in the oceans. They're the base of all food webs. What I mean by that is microbes can be food to other organisms. Many invertebrates eat microbes, as well as playing a major role in nutrient regeneration and supply of nutrients. And of course, they support all plant, animal, and human health. So bacteria are my friends. They should be yours too. Here's a few kind of, I would call them cocktail party facts, if you want to, that's, you can like quote these numbers and things, next time you want to show your chops about the microbiome. There's thousands of microbial species living on every epithelial surface of the body, in and out. And in fact, I can't even make that statement anymore because there are scientists that are showing that on your skin, so I always thought microbes are just growing on my skin. It turns out that there's strong evidence that within your skin are microbial populations interacting with the immune system with these dendrites, okay? There's a lot of discussion going on right now about whether the placenta in a pregnant woman has an associated microbiome. Okay, so the old story was every epithelial surface has an associated microbiome. We may be limiting ourselves by making that statement. But in fact, most are living on epithelial surfaces. I tried to come up with a quick way of describing it, so I'm showing you different body regions, the general surface areas of those body regions, and the general densities. You can see they're incredibly numerous. Okay, and there are many. Genera is, for those who are not biologists, just a way to categorize microbes. So it means that so species is more detailed than genera, so that's just genera. Under there are thousands of species, okay? And collectively, we've coined the term microbiome to mean this whole collection of microbes that live in and on our bodies. I mentioned breast milk. That's something that's kind of new. Turns out there may be, in fact, a breast milk microbiome. What it's doing there, what contributions it makes to the offspring, all those are yet to be known. We acquire these microbes every generation. Okay, so I'm having a great conversation with Margaret this morning over breakfast, and she taught me a term, facilitated horizontal transfer. I thought it was vertical transmission because of the mechanism, but in fact, she's corrected me, so I'm going to be clear. Okay, facilitated horizontal transmission. But basically, there is a process, a very important process, two processes actually, that occur that nature meant to guarantee this transfer of microbes. Number one, when a woman is pregnant, her vaginal microbiome, the microbes that live in there, start to change. And the beneficial ones become enriched, and the ones that don't really do much drop off, okay? So when the baby's born vaginally, so through the birth canal, there's a lot of goopy mucus, right? And there's this kind of, is somebody laughing? Am I grossing out? You know what? I give talks and I gross people out all the time. So, you know, you can laugh, you can go, ee, or whatever you want to do. Just go right ahead. But I'm going to go ahead with my talk. The mucus is now laden with all these beneficial microbes, and here comes the baby, and it gets coated in the mucus, okay? It's a vehicle. It's a vehicle for transmitting an initial inoculum of microbes to the infant. The child is what we call microbe magnets. They're meant to acquire microbes. Those of you who are parents and you send your kids to daycare, you know all those microbes they acquire. Well, you don't like that part, but it's part of a natural mechanism. They're meant to pick up microbes because that's essential to and I'll get to that in a minute, essential to the development of their own immune system, okay? So these millions of genes, which I just mentioned, encode this myriad of metabolic capabilities. These are my cocktail party facts. The gut microbiome in an adult weighs about the same as the brain. So you got 3 pounds of microbes down here, you got 3 pounds of brain up here, and it has the metabolic capacity of the liver. Okay? And in fact, the human microbiome augments, extends, and supports those capabilities encoded in the human genome because it's a paltry number of genes at 23,000. Now, my home at NIH is the National Human Genome Research Institute, and I cannot tell you how many awkward conversations I have had with my colleagues because, of course, I'm the bug lover, and they're the human genome that does everything. I don't know that I've convinced them all, but I have begun to turn some of my colleagues around on this idea. But it's true. We know now that the human genome is about 20 to 25,000 genes, and yet that's like a paltry amount compared to the millions of genes. And evolution, it's like a partnership. Well, you need this, I'll come along and give it to you. The human body in an adult finally matures into an ecosystem of microbial habitats. This type of data here is called a PCOA plot, and it's a way to collapse really complex data into a visual way to be able to think about it. So each one of these dots, okay, is a sample taken from that body site, whether it's GI tract, oral, nares, skin, or urogenital. The DNA is extracted and sequenced and analyzed, and then we try to compare between habitats or between people. So one of those dots is an individual and their GI microbiome, another individual and their oral microbiome, okay? And what I want to show you here, without going into the statistics, is that the GI microbiome and the oral microbiome are as different as Hawaii and the continent in terms of the makeup and complexity and diversity. And in this audience, our gut microbiome, all of us, is more alike than my oral microbiome and my gut microbiome. I might as well be two different continents, but here we're like one collective of islands, I guess. And the microbiome and host interact. This is a highly co-evolved system. They interact to help digest the indigestibles in the host diet. And in fact, they ferment the plant material to produce their own energy sources. I just want to show you on the right. In fact, I'll stop for a second. Let me just show you on the right, because I find these images, I'm a very visual person, so I find these images are amazing. At the top is a gut cross-section. The blue are the gut cells, so your gut lining, okay? And then there's this tiny 50-micron thick layer of mucus that has to be sustained because, in fact, we have kind of a microbial détente with our microbes. So if they enter and touch the gut wall, then, in fact, they can start to penetrate and cause problems. So we really need that gut, that mucus layer. And then that sort of purple and pink area are all kinds of different gut microbes. Below is a similar colorized micrograph of an oral biofilm. So this morning, you brushed your teeth and you had some schmutz here in the corner. If you were to take that schmutz and stain it, it would be this gorgeous thing full of all kinds of wonderful microbes. And do you know that in your mouth, there are at least 9 microbial habitats? Right now, I'm talking to you, and my tongue is slapping the roof of my mouth, but the microbes on my tongue are totally different than the ones that are growing on my palate and on the roof of my mouth. And the microbes that grow right here at the tippy edge of my tooth are totally different than the ones that are growing right there under my gum. 9 different microbial habitats and 9 different communities just in your mouth. So they help digest the indigestibles. They help educate the immune system to recognize self from non-self. There's a whole separate lecture I could give about how the reason that babies are meant to acquire microbes is they don't have a developed immune system yet. So as their immune system develops, those microbes become part of this is part of the immune system. These microbes belong here. Type of communication that happens in the body. So self from non-self. They produce energy sources for our host cells. Specifically, the best example I can provide is, you may recall from high school biology that most of our cells use glucose, but not in the gut. In the gut, short-chain fatty acids like acetate that are fermentation byproducts from fermentation that happens in your gut is the major energy source for the gut cells. Okay, that's a highly evolved process. They use a totally different energy source, and they rely on microbes to supply it. Microbes produce all manner of beneficial compounds to their host, like vitamins and antimicrobials, and there's a really hot area of research in which it's quite clear that the gut microbiome is communicating with the brain. And also gut microbiome communicating and affecting the mind. A lot of neurotransmitters, for whatever reason, are made down here in the gut. I don't think we entirely understand what the communication links are, but there's a lot of information that shows us that the gut microbiome and their activities affects brain development and behavior and mental health and so on. Okay, but so what's also come about is another field looking at disease transmission and so on, as a result, obviously over here to the left, as a result of use of vaccines and antibiotics. In most cases, not entirely, but in most cases, we've managed to actually decrease the incidence of infectious disease through the use of vaccines and antibiotics. But what epidemiologists have also seen is this kind of mirror image explosion of allergic and autoimmune diseases. You know, what's going on there, they're asking. What are we doing that we're somehow causing this extraordinary increase in a huge array of allergic and autoimmune diseases? So what's causing the appearance of these chronic diseases? Okay, and then another group of scientists are asking another kind of question. There was a beautiful paper written in 2009 in which two scientists postulated and were concerned that in fact there may be the systematic loss of these microbes by generation because of various practices, things that we rely on, things that we think are good. Okay? Look, I like clean water. You know, I don't know what else I like on that list, but I like clean water. But many of us live indoors, are not exposed to the outdoors. Cesarean birth, which short circuits the transfer of microbes to the baby. Antibiotic use and overuse, low fiber diet. You know, most of us don't even meet the USDA requirement for daily fiber intake. And why do we care about fiber? Because it's the food for the microbes in our gut. So if you starve your microbes, they're not going to be happy, and some other microbes are going to take over. So low fiber diet, processed foods. The reason processed foods are a problem, they're kind of, to my mind, the way I think about it, they're kind of equivalent to food that's already been partially digested. So not much gets down to your large colon. So you're kind of starving your microbes if you eat processed foods because it's too easy for your own body to digest. I'm learning recently about all manner of food additives being a problem. I think one of the big ones recently is emulsifiers. Okay, if you eat like ice cream or chocolate milk or peanut butter or whatever, they always add emulsifiers because they want to stabilize the food. You don't want it to separate in the jar or can or whatever. Well, what's happening with emulsifiers apparently is that it's causing this, you remember when I told you there was this 50 micron mucus layer, and actually the microbes play a major role in maintaining what's called the tight junctions between the cells in the gut. Well, emulsifiers are bypassing all that and allowing gut contents to move outside the gut. So if you eat emulsifiers, apparently you're short-circuiting that entire process, and stuff in your gut, which should remain in your gut, is leaving your gut and going to other parts of your body. And apparently lupus is a kind of disease that suffers from the presence of leaky gut or gut contents leaving the gut and moving to other parts of the body. So all these things, which in most cases we think are good, are having unintended consequences. So these two scientists are saying, with all these different practices, are we having this sort of systematic, generational impact on the richness of the microbes that live in our bodies? Okay, the title here is Microbial Diversity Does Appear to Be Decreasing Across Populations and Across Generations. And I think this will be particularly important to this Hawaiian community. So here what they've done is taken, 24 different populations from around the world and studied the diversity of their gut microbiome. But they've been grouped into 3 kinds of populations. At the very top, the top 6 or so live in jungle or savanna and are hunter-gatherers. Okay, and they collect their own food. The people in the middle, rural, they live in rural populations. They may be farmers, but they live in rural populations. And then the bottom dozen or so, starting with Japan and ending in Ireland, are all what we would call modern societies living in urban environments. Look at that trend, amazing trend, and drop in microbial diversity across these populations, possibly due to these lifestyles. I think that's what we're trying to hypothesize. Over here, loss of microbial diversity over generations. This is an example where they looked at populations who live in I think this was Vietnamese maybe, I don't remember right now, but somewhere in Southeast Asia, Okay, so they measured their microbial diversity in the gut before they immigrated. Then they immigrate to the mainland. They didn't talk about Hawaii, but they immigrated to the mainland. And within a very short time, they started to see a change in the gut microbiome, and they're starting to gain weight. BMI is increasing, okay? Then after they've lived in the US for a while, long-time residents, their BMI goes even higher, their microbial diversity even lower, and then the next generation of that immigrant population has a BMI and low diversity like European Americans that have been here generations. It only takes 2 generations to totally radically alter one's gut microbiome when you immigrate from wherever your home country was to come to the US. Okay, so that's what Stan Falco and Marty Blaser predicted in 2009 has happened. It is happening. So here's where the Human Microbiome Project comes in, and I think a lot of my slides coming up are going to be full of like graphs about budgets and numbers and things like that, because I'm trying to sell it. But so the thing was that a lot of these ideas were coming up, so NIH listened, and they said, you know what, we have an opportunity to contribute to this by developing a toolbox of research tools to allow the research community to explore these questions they're concerned about. And I call it timing and opportunity because, in fact, at the end of the Human Genome Project, there was a lot of capacity. Genome sequencing centers were available. They had just announced the human genome had been sequenced. There was a lot of technology that had improved, so they were more affordable, and the cost per genome was dropping like crazy. So that's 2000. You can see the cost was very high, and right about the middle of the 2000, you start to see this precipitous drop in the cost of sequencing. And the NIH was concerned about cost because it would, even though it was super expensive to sequence the human genome, based on what the microbiology committee was telling NIH, they were kind of flabbergasted. They were concerned they were just going to go bankrupt trying to sequence the microbiome because of the diversity and complexity. But they realized with the drop in sequencing, they could afford to do it. And also another timing and opportunity thing was NIH is amazing, is a major funding agency. It's got a $30 or $32 billion per year budget, but it's got 27 institutes and centers. So they take the human body and they cut it up into 27 different components, right? And they're all siloed, right? And of course, as you might well imagine, there's also a lot of gaps and redundancies, right? So Congress says, we're not doing this. So they created something called the Common Fund under the Office of the Director in order to ask NIH to look for things that are trans-NIH interest. And they had a budget to be able to fund those things. So the Human Microbiome Project was funded under the Common Fund to sequence what they called the other genome. That was the other kind of catchphrase of the microbiome, was the other genome, right? So the Common Fund's first project out the door was the Human Microbiome Project, and they devoted $215 million and 10 years to build this research toolbox. Our job was, here's a brand new field, what do these scientists need to get started? And that was our entire mandate. So as I mentioned, in the 70s, we started developing tools to directly study microbes in nature. Now, this is the next phase of studying microbes in nature. We're not just looking at them under the microscope, we start studying them, studying their DNA. So we have the ability, I already mentioned, to use specific markers to look at bacteria in samples. We also started to develop the capacity to sequence single microbial genomes, but maybe most importantly, it's called metagenomic sequencing, because it's multiple genomes. We were able to now begin to analyze total microbial communities, as well as the development of other high-throughput technologies to measure the metabolites, the gene expression, and other major biological properties of microbial communities, including these apparently brand new discovered microbial proteins. So HMP had two phases. Again, remember, we're building a toolbox. Okay, the first phase phase was to try to characterize microbiomes, so the tools and the approaches for that were developed, and then see if they correlated with phenotypes. And it was a kind of a naive approach. You know, if a person is healthy, do they have a characteristic healthy microbiome? If a person had type 2 diabetes, do they have a type 2 diabetes microbiome, and so on, okay? So we had this amazing, what we call the benchmark healthy cohort study, in which 300 adult men and women were verified to be free of disease by clinicians before enrolling in the cohort. What do I mean by that? When I go to my doctor for my annual exam, I get my blood drawn, I donate some urine, he checks my blood pressure, you know, a few things like that. I'm talking about a clinician verifying that I had no skin disease, another clinician saying I had no GI disease, and so on and so on and so on. And even though these were young people on the average age of about 25 or 26, 80% of these Americans had oral disease. Okay, and we had to raise the BMI for entry into the cohort because nobody could meet that lower BMI level. So then we had to send all these people out to their dentists and periodontists and whatever else to get their dental caries, their gum disease, and everything else repaired, cleanse the antibiotics from their bodies before they could re-enroll in the cohort. So even though we're going after a healthy group, it was hard to find healthy. It was hard to find healthy. Okay, so we analyzed the 5 areas, gut, skin, nares, mouth, and urogenital of these 300 people to really ask the question, who's there, and does it correlate with phenotype? And the answer is, no, it doesn't. It doesn't. You cannot just name the microbes, you know, with a sequence analysis and say, yes, I know what health status that person is in. And I'll give you a tiny, tiny little example of why you can't just name the microbe. Okay, there's a microbe that lives in our body, in our gut, called E. coli. You've heard of E. coli. Everybody's heard of E. coli, okay? You've probably also heard of Taco Bell E. coli, okay? It's still called E. coli. One of them will kill you, okay? So you can't just name the microbe to know what it does. And yet we set out asking that kind of question and found, in fact, we couldn't stop there. And the demonstration projects were for diseases, like we had these 12 demonstration projects to see if we could demonstrate if there was a characteristic microbiome associated with disease. So, you know, I think it was one type 2 diabetes cohort, one preterm birth cohort. We had a bunch of these. And so we developed the tools for sequencing the microbiome, but we realized we couldn't stop there because you can't just name the microbe or characterize the microbial community and get at the condition of the person. So we had a phase 2. Okay, this one was, what are they doing? We already know we can't just ask who's there. We also have to know what they're doing. So there was an entire expansion of other properties that were collected from 3 different cohorts. And we also track the host properties over time of a type 2 diabetes cohort, an IBD cohort, and a preterm birth cohort. And even if you don't care about those conditions, the point was this was a rich data set, carefully vetted. All of the data were there and available to anybody who wanted to sit and analyze the data. They were reference data sets so that anybody who wanted to jump in and look at it can kind of do like dry experiments without spending the money. I mean, that's a real bonus for a lot of scientists. They don't have to spend the money. They can test some of their ideas in a carefully vetted reference data set. And so all those data went into our HMP Data Analysis Coordination Center. You can go there, you can download data, you can look at the protocols, you can look at the pipelines, you can look at the computational tools, you can play around with the data. There's all kinds of things you can do. So we met our goal of trying to build out this toolbox. Don't worry about that. Oh, I do want to talk about this. And at the same time, even though it was not a deliberate goal of the HMP, we also created a research community because we functioned as a consortium. We had a bazillion conference calls and meetings, and we're always talking about QCing this and referencing that and round-robbing this and so on. So there was a lot of interaction across these members of the HMP consortium, and that actually launched the research community in the US because so many were involved in the HMP. Okay, here comes the numbers. So these are for like the upper administration and other people who care about funding. Now, we built a toolbox. Did anybody care? Well, here is what captures that it would completely democratize human microbiome research in this country. Because the data on red is the HMP data per year, starting in 2007, the budget. Okay, then I figured out a way to find the budget for the other non-HMP human microbiome projects, and look at that explosion of support. Okay, so that total over 10 years totals $880 million. So we catalyzed the field by providing the toolbox in the beginning, and then people jumped on board and were able to use the tools to ask questions about their own disease of interest. Okay, and I tell you here, these are 1,000+ studies. I read the proposals, I read the studies. All of them reference the HMP reference data and tools as the basis for the study of the microbiome in their disease of interest. We completely succeeded in providing the toolbox that was needed. And I told you there are 27 institutes at NIH. Currently, 21 of them fund in this area. In the beginning, it was like 5. Okay, and we published a paper on this deep analysis of what was being funded in this area at the NIH, if you ever care about this kind of thing. And here's sort of a cartoon of the different kinds of diseases that are being studied. I just show you here in the corner. There's sort of a close-up of the last 5 years of that 10-year window. A total of $728 million. 2/3 of it was on disease. But for those of you who care about basic biology, a very healthy 1/3 was on basic biology. Nothing to do with disease. Because it's such a new area, there were so many fundamental questions we had about the microbiome that a very healthy $262 million over just a 5-year period was devoted to the basic biology of the microbiome. Okay, 100+ classes of disease, and these are the World Health Organization category of classes, ICD-10. 100+ classes of disease are being studied at NIH. Name it and it's probably being studied. Okay, and this is just a little snapshot of it. And I use the word association because it's too soon. We don't have cause and effect. So there's no yet evidence that a microbiome causes disease, but there's some kind of connection. Something's going on with changes in the microbiome that is somehow exacerbating or somehow interacting and causing disease. And here are some surprises. Type 1 and type 2 diabetes. Type 1 diabetes surprises me. Lupus surprised me. Multiple sclerosis surprised me. Even autism surprised me. There are old ideas about the role of microbes in cancer. That whole idea has been completely resurrected by the National Cancer Institute. So there are many, many kinds of cancers that are being studied and with regard to the role of microbes in those cancers, GI tract diseases, heart diseases, and so on and so forth. And it kind of makes sense, right? If you got this central thing happening in your body and it's disturbed, you start seeing all these impacts across all kinds of health conditions. It's not a big surprise that we're looking at 100+ classes of disease. I mean, it's an eye-opener, but when you stop and think about it, it's not a big surprise. Okay, now what I want to do is transition to environmental microbiomes because I don't want you to walk away from this discussion, from this lecture, thinking, oh, well, you know, it's just about humans, right? No, it's not. There are scientists studying the role of what we call atmospheric microbiomes. Aeolian transport, you know that term? It's like global winds that pick up material from one continent and carry it to another. There's aeolian transport of microbes from the Saharan desert to the coral reefs, including fungal spores that probably are causing fungal disease in coral reefs. Warfighters, the Department of Defense is into the microbiome in a big, big way. And soldiers are now called warfighters. That's the new term. Because they're deployed all over the world, they're highly stressed, they're introduced to a different diet, and they lose a large fraction of their warfighters to sickness. So the Department of Defense is trying very hard to develop healthy diets, gut-friendly diets, and things like that for their warfighters so they don't lose so many soldiers before they even before they even engage in whatever it is they're there to do. livestock and poultry. Most of you know that in our country we have, I don't think the term is monocultures, but single strains of cows that we raise for dairy or meat or whatever, and single kinds of poultry, and we dose everything with antibiotics because we have close quarters for all these livestock. We have completely ignored that all these animals have their own natural microbiome, and now there are some communities that are trying to find a way to rediscover, for example, the ruminant microbiome in cows, and seeing how can we better support that to have healthier livestock. Soil and crop. It's even worse with plants, with crops. You know, we grow monocultures of corn or monocultures of wheat or whatever, and they've been bred so much that they've lost their natural root microbiome that they live with in the wild strains. So now there are agronomists trying to rediscover those natural wild strains and see if we can rediscover the natural microbial communities that live with them, because plants don't move, right? It's kind of a weird lifestyle. They don't move. And so they're constantly bombarded with all kinds of, you know, germs and infectious agents. And in fact, what happens is they nurture a very rich microbial community to help them fight disease. But we've removed all those microbes. They're still standing there in the soil. And so what we have to do is dose them down with all kinds of pesticides and stuff so they can be protected. So now we want to rediscover the natural ones. Coral reefs, you guys already know this, in Hawaii, they're dying like crazy. There's a natural microbial community associated with coral reefs, but they're highly stressed, and we need to find a way to restore coral reefs by also learning better about their microbiome and their defense mechanisms. You may not know this, but NASA's into the microbiome in a big way, because supposedly we're going to go to Mars or something, I guess. Maybe Elon Musk is going to send us to Mars. But the point is, with the International Space Station, they've learned a lot about the gut microbiome. It turns out that the longer an astronaut stays up in space, even though they live in microgravity, two bad things happen. Their immune system gets weaker and weaker and weaker, and at the same time, virulent microbes become more virulent. So it's a kind of a double whammy. And so they did that experiment with the twin astronauts, and I don't remember the one that was up in the, was it Mark Kelly that was, anyway, he's not in good shape because he was up there for a year. And so they're trying to figure out, how do we fix that? If we're going to go to Mars or the moon or whatever, I mean, people are going to die along the way unless we can find a way to make sure that their immune system is more robust and remains robust. And that includes the microbiome, because Mark Kelly's microbiome went haywire too when he was up there for a year. Oceanic, well, I mean, that's a given, right? I already told you that of every 2 breaths you take, one of them comes from the oceans. They're also a major carbon sink, you know about that, if you read kind of even casually about global climate change. You know, the oceans are the lungs of the planet, right? And the most populous populations of microbes are in the oceans. So we really got to make sure we understand what they're doing and what they need to maintain their most robust abilities. And finally, because we live indoors all of our lives, it turns out that, and people have done this, you can go to someone's home and you can figure out who lives there. Because we shed, I wish I'd remember the number, we shed an extraordinary number of skin cells in a 24-hour period. We also exude a lot of microbes through breathing, through movement, everything else. So our indoors, if I were to swab this entire auditorium, it would be laden with microbes because we've shed them. So the reason that even matters is because we have a modern indoor lifestyle and we shed so many microbes so that we actually create indoor microbiomes. And it's particularly important in places like hospitals and daycares. So they're trying to figure out, how can we change the architecture of hospitals and daycare centers so we can reduce the potential burden of microbes in those environments, if we, so that's another area. So these are all areas outside of just the human health area that I talked about, in which the field of microbial sciences is making a major, major impact. So the White House, I had the great privilege, wow, during the Obama administration, there were so many wonderful things that happened, so many wonderful things that happened. You know, I feel like it was a dream. But I had a chance to work in this White House Office of Science, Technology, Policy, and it was a committee that was formed. Of course, it's a government, it's got to be a committee. It's called the Fast Track Action Committee, Mapping the Microbiome. And we were chartered and required to collect data, and these are all the different agencies that were involved, okay? To collect data, the question was, how much are we really funding in the microbiome area? So they farmed us out and they said, go collect that data. And in fact, we identified over a 3-year fiscal period almost $1 billion, not just in human microbiome, but almost $1 billion of research in the microbiome. Of course, NIH was a very large fraction, but look at Department of Energy, National Science Foundation, look at all the agencies that were involved. Okay, so many, many federal funding agencies across the entire U.S. government has been and is funding in the larger microbiome area. And it's occurring in lots of different habitats. We identified 8 systems: agriculture, aquatic, atmospheric, built environment, human, energy, non-human lab, that means like animal models, and terrestrial. That $922 billion was apportioned this way, about 1/3 to human, 29 to animal, and so on. But the point was, it's not only human microbiome. There's rich research going on across many, many different systems. And that excited the White House. You know, you folks may not know this, but President Obama is a complete science nerd. He loves science. And it turns out he got completely turned on about microbes. He gave a State of the Union address in, oh gosh, I wish I could remember when, 2014 it was, and where he used the word microbe twice. You know, really, I have a copy of it. You know, he used the word, he used the word microbe twice in a State of the Union address? Oh my God. So he was very gung-ho about this. He was really excited about this data. So a national microbiome initiative was launched, and the reason that you should even care is, you know, the federal government's a big behemoth and a lot, you know, it moves so slow and can never get the different parts to talk to each other. But when you create an initiative, what it does, it gives us permission to collaborate and coordinate. So all these federal agencies got together and created this another committee, right? Microbiome Interagency Working Group to develop a federal plan. Okay, here's this amazing new area with all these implications. What can we do? Well, we can certainly try to coordinate. We can develop new technologies, and more, maybe most importantly, it's time to build the microbiome workforce. We have what we need. We got the tools, the questions. Now let's just educate the next generation. But I will tell you, I'm going to give you a slight downer. This happened in 2016. Guess what happened after that? Okay, and so we've kind of gone to ground. But we're working, but at a kind of a, you know, underground level. So even though the National Microbiome Initiative is there, we can't. So there's some things we can't do. That chartering by the White House, it's a real formal process. It has to be like approved and stamped and agreed. You know, he has to sign, or she, whoever the president is. So. But you know, we still have our connections and all that, and we're doing certain things. Okay. So for example. The Phytobiome Initiative was born out of this excitement out of the National Microbiome Initiative. This has to do with trying to understand, you know, the plant-soil microbe interactions, kind of basically going, I think agriculture is going through a renaissance because they're rediscovering soil microbes and the fundamental roles they're playing. So this Phytobiome Initiative is a grassroots organization basically trying to find a way to develop networks across the research teams that are working in this area. They've birthed a journal, they have an alliance, and so on. DOE, even though there's not a National Microbiome Initiative, they've gone ahead and decided they're going to try to develop another resource, not a toolbox per se, but another resource for the environmental area. And it was a takeoff from the HMP. HMP has built a toolbox, so now it was time to build a toolbox for the environmental field. And it's called the National Microbiome Data Collaborative. I'm on the Science Advisory Board for this group, but they've gone ahead even though there isn't an NMI, so that's very heartening. And what's really amazing to me, again, this is grassroots. Last year, we identified about 3 dozen universities around the country that had microbiome groups. That maybe their deans or their provosts or whatever birthed, gave them some seed money to get it going. But there's so many of them, the question was, well, shouldn't we be trying to work together? So there was a meeting at Irvine. And they agreed they wanted to form a Microbiome Centers Consortium as a way to work together. You know, let's not reinvent the wheel here. Let's learn from each other. So in fact, Simaiki is a member of the Microbiome Centers Consortium, of course. Of course, thank you, let's clap. Indeed. And it's an opportunity to basically work together across universities, also across fields, because I write in there, not all focused on human. Many of these centers have no scientists working in the human microbiome. They're all working in environmental or symbiosis or something else. But the point is, we're still asking the same kind of questions, right? What are these microbes doing? What are they providing? How are they being perturbed? How can we restore them? All those kind of things. So we want to go across disciplines and ask those kind of questions. This is a beautiful thing. Oh, we wrote a paper recently to invite other universities to join the consortium, so that's what it's a discussion about the need for it and inviting other universities to join. Oh, and this was my little cartoon of trying to figure out all the connections. So you can see all the different topics that are represented by this consortium of microbiome centers. Some of them might sound kind of funny, and I'll call out one, for example. What is microbiome law and ethics? What do you think that is? Well, I'll give you an example. Okay, we're laden with microbes, right? I go to the bathroom, I flush the toilet. I have now said by flushing the toilet that I have no need to keep that. Microbes go down the toilet, somebody goes out. I have recently heard from a colleague that we have a company that goes to the airport and collects the bilge water from airplanes because they figured out it's the most diverse microbial material they can get their hands on. And what they want, wait. They're gonna make buco money from that because they're gonna isolate microbes from it, they're going to get new pharmaceuticals, they're going to do all kinds of things. So I flushed my poo down the toilet. If they make $1 billion product, what happened? Where's my, you know what I mean? So, I mean, and there's all, and also, I mean, I could talk about law and ethics on the microbiome forever. But the point is, there's these other kind of shoot-off fields that are developing around the microbiome that you might not think of at first. So there will be lawyers in the next generation who will specialize in the microbiome, for real, for real. Okay, and I want to make another remark, okay? Do you remember the graph on the right? That's the one where I showed you that the HMP catalyzed human microbiome research at the NIH. But I also I put in that yellow block because I wanted to show the shift, okay, between the growth of microbiome centers and the NIH funding. There's a lag. And the reason that's important, that means that we're just begun growing microbiome research, just begun. Even though $880 million sounds like a lot of money, no sirree, Bob, it is not. This is just growing. And not only on human. And I want to come back to Hawaii. I don't know if the audience knows this, but Hawaii and University of Hawaii has an absolutely amazing legacy when it comes to microbiome science, microbial ecology. Because starting in the 1980s, there was a long-term monitoring station established in the Pacific Ocean called Station Aloha, and there has been continuous, non-stop monitoring of those marine microbes since 1988. Counting, yeah, exactly, isn't that, who said wow, isn't that amazing? Yeah, I mean, it is amazing, you know. Come hell or high water, there was no stopping that continuous monitoring. And then there was another one established in the Pacific because, I mean, the Atlantic, because we want to have these long-term two monitoring stations in the major pelagic bodies surrounding the continental U.S. But the point is, those data now are feeding into climate models because we have this data, long-term data, that's unparalleled. This happened in Hawaii. So what I want to point out, exactly. Well, I want you to clap about this too, because now we have the next amazing thing that's going to happen with the COBRA activity in the Integrated Center for Environmental Microbiome and Human Health and CMAICI. They are building on the legacy of Station Aloha and all that meant to the scientific community, and now launching it further. So Hawaii is the home of microbiome science. I mean, it's not an exaggeration. You have an amazing legacy here. Thank you.
Have you done, or can you envision doing, as we're having a steadily aging population, a microbiome study on the healthy gut for someone who's over 65.
Yeah.
I noticed you chose a cohort of 25-year-olds. That's great for them, but there's a lot more of us right now that are going to be rather older, and I'd like to know what to eat to keep that going. Thank you.
Okay, so I can very quickly address that. I mentioned there are these 27 institutes. For sure, there's a National Institute for Aging. Of course there is, right? So NIA, National Institute of Aging, is doing an elderly microbiome. However, if you want to read more, there was a very important study done in Ireland called the ElderMet study, or Elderly Metagenomics, by the University of College Cork, Ireland. Okay, and it was started probably in 2006 or 7, and it went on for about a half a dozen years. They did an amazing study of their elderly, which I believe they defined as 65 and over. So they did kind of the baseline landmark study, but now there are other elderly microbiome studies ongoing. There's even little experiments with animals where they're doing things like transferring the gut microbiome from a younger animal to an older animal to see if they can sort of revive it. In fact, it does work. So maybe that would be the way you kind of rediscover the fountain of youth, is have a fecal transplant. I don't know. I don't know, but there's work being done in other populations. Yes. Other questions?
What do you see as the best tool to use to understand the handshakes between the microbes and what they're doing?
Good question. I fear that we didn't do a good job in that part of the toolbox. What we did is we developed a lot of metabolomic. So what's metabolomics? You know, there are a lot of molecules in your body that are circulating through your body, right? And they give us a lot of information. You know, your doctor measures cholesterol, for example. That's one molecule that's used as an indication of a health state. So we've been trying to use metabolites and measured by a procedure called metabolomics to see if we can get an insight into that handshake between microbes and host. But some of the tools are not ones we did a very good job of developing. That's happening more in the immunology field, where they're trying to develop methods for looking at the crosstalk between the human system and individual microbes. So that's an area yet that still needs more development.
Right here, but the mic does not. That's fine.
Yeah.
A very basic question. So, given your own understanding of these different microbes and your own experience with different microbiome projects, how would you define a human being?
Oy, oy, oy. You know what? That is actually an area of active discussion. A human being is...
Your own definition.
Oh, okay. So I can take off my NIH hat? Okay. A human being is an ecosystem. Okay, what's an ecosystem? It's a collection of different organisms working together towards, let me maybe make up my own definition. It's a collection of organisms working towards a common good, if you will. So in this case, the common good is trying to maintain this thing you see in the mirror, okay? So I think, yeah, maybe the human is an ecosystem of their attendant microbes and the thing that carries them around on the planet. Is that okay? Okay.
Good segue to a UH scientist.
Yes.
Right. This doesn't work for some reason. Well,
Here.
we go.
Oh.
Thanks, Lita. Aloha. Oh. I don't know how am I supposed to follow that, but can you please all join me in thanking Lita for that wonderful talk?
Oh! Welcome. Thank you.
So I think there's a burgeoning area of obviously the microbiome, and with that, there's many faculty here who have a increased interest in incorporating that as part of the work. So some examples I'll give you, and we're tasked, very difficult task actually, for those of you who know me, that we were given 1 minute each to talk a little bit about our work.
Oh, nice. Okay, great.
For those of you who know me, that's impossible.
Yeah.
But some of the research that we're working on right now is actually trying to understand what the microbiome in the gut how that relates to health. And in particular, we're actually doing the first study ever in Hawaii where we're looking at the context of the social network, not Facebook. The social network, so families and friends that influence you. We're actually working with a community out in Waianae, Manoa Organic Farms, who takes in interns as part of their program and looking at the impacts of their program as they are actually exposed to the environment and health and health decisions. And obviously the program incorporates a lot of education, and so that how that influences the gut microbiome composition and how that relates to resilience factors of health, and also to potentially reduce risk for certain chronic disorders that we see in these communities at a high incidence. So that's one of the many studies that we're trying to understand the relationships between the microbiome and the human condition. But actually, I mean, if you think about, for those of you who don't know me, I'm from Waianae. I was born and raised in a 4th-generation household with very indigenous family. And my great-grandmother taught us La'olapa'au practice. So how the environment connects to health is a very important concept that we all learned. And actually, the practices that were in place in Native Hawaiian culture actually, I think, had an understanding of the relationship with aina-based practices to the environment and how that supports health, and that included the microbiome. So as a short anecdote, for those of you who know poi, right, and pa'iai, so if you think about it's taro, for those of you who don't know what that is, it's pounded with a pohaku stone, and it's actually it inoculates the microbes on the stone because it's never sterilized. And that the way that poi was eaten was a communal situation where you share the single bowl of poi and with your hand, around the table. So the fermentation process of the nutrients, obviously, of the kalo was an important consideration for supporting health. And so I think the practices and concepts from indigenous cultures, especially in Hawaii, understood that these environmental factors, including the microbiome, can be supportive of health in that ecology. So I totally agree with you that we see ourselves as not just humans itself, but as a part of that ecosystem. So mahalo. We have another example from other faculty who are interested in looking at the microbiome. Minjin, you want to come and explain some of the work that you're working on?
Thank you, Alika. So my name is Dr. Minjin Lee. I'm a practicing maternal fetal medicine high-risk obstetrician here at Kapiolani. And one of the programs that we're doing right now that is sponsored by the Ola Hawaii program is actually doing what we are calling a gum-to-bum analysis of the pregnancy microbiome for looking at how diet and nutrition affects the microbiome over the course of pregnancy, from mother to fetus, and then the baby as it's after birth, and looking to see if changes and the differences in the diet and nutrition that women ingest associated with any kinds of pregnancy outcomes, diabetes, premature labor, growth restriction. And so this part of the program that we're working on at Kapiolani is trying to map this interaction from the mother to the baby and looking at the various microbiomes that can affect health and disease.
Mao.
Thank you. This is all really fascinating. I'm really interested in all this stuff. So I have a question. You mentioned, are we good? Okay. You mentioned there hasn't been a strong correlation between the microbiome and the phenotype when it comes to someone's health. So I wanted to see what your take is on all of the health claims that we're hearing now about eating fermented foods, specific microbes like Acidophilus and Bifidus being beneficial to health. Are those real? I mean, can we really change our microbiome by the foods that we eat? You know.
So that's quite a large question. I have a mic. First of all, I want to clarify something because I don't want to misrepresent it. I probably was a little too fast when I was talking. What I was saying is you can't just name the microbes. We thought we could just sequence stuff, name all the microbes in a community, and that it would be clear as day that it's associated with a specific phenotype. We thought that way with disease. You just sequence these sick people's gut microbiomes, and sure enough, it's going to be clear as day that person has diabetes. Isn't that turning out that way? Because of that little joke I made about E. coli. You can name the microbe, but you can't tell us what it's doing, okay? Because they have an incredibly versatile metabolism, and depending on the environment, they'll express a different metabolism in this environment versus this environment. You know, Ned Ruby works on what's called a facultative anaerobe. It breathes air, and it also breathes other compounds. So you can name the microbe, but you can't tell me what it's doing. It's going to be habitat dependent, environment dependent. So I want to make that clear. So what happened was we thought we could do it was a very medical approach. We thought we could just sequence this microbiome and it would automatically tell us about the health state of the individual. However, the other thing you said was there's no connection between the microbiome and health. Absolutely, but we're not going about the right way. We have to look at the interactions between the microbes, what they're doing, and their metabolic features, what they're doing in the system. I want to make sure I understood your second part of your question.
So there have been certain.
The other one that was asked. Okay, that was asked of me at the Kapiolani. Same kind of question. Here's my take on it. Okay, I'm going to take my NIH off. Okay, here's my take on it. Yes, when you eat, you should eat to feed your microbiome. Okay, so when breakfast in the morning, you think, okay, what would my microbiome want? And feed it. Lunch, what would my microbiome want? And feed it. Not what Lita Proctor wants, right? But that's what I think is probably a good principle to operate under. And be consistent. You know, you see all these blessed food fads, right? Don't jump from keto to paleo to whatever else, okay? Just stick with it. Be consistent. Eat lots of fiber, but don't do it all at once because you're going to get really sick. Do it slow. The USD recommendation is it 15 grams for women and 20 for men or something like that? That's about half. You need twice as much as that, okay? I don't know how they came up with that number, but you need to ramp it up way past that 15 grams per day, and it can't be in the form of fiber pills. Okay? It has to be in whole food. It has to be in fruits and vegetables and grains and beans. Don't forget the beans. They provide a lot of good fiber and other needs. So I guess I'm not sure I'm doing a really good job following that kind of a diet, but the point is I do try to pay attention to that. I think it's as simple as trying to feed your microbiome. Now, they sell, I'm going to do a little rant about probiotics. Is that okay? Don't buy probiotics. Don't buy probiotics. Now, the principle of probiotics is probably sound. There probably can be, in the future, products that you buy from the drugstore that has a specific microbe in it that can restore a function in your gut microbiome. But they don't exist today, okay, because probiotics are not regulated. You know, you don't buy drugs from the grocery store. You buy them you get them as a prescription from your doctor. You want a regulated product if you want to treat a health condition. So probiotics, number one, are not regulated. And we have colleagues, all of us have colleagues, that have gone to the drugstore and sequenced that stuff that's in a probiotic bottle. It's either dead, it's either contaminated, it's not the microbe they say it's on the label. And they're expensive. So don't spend your money. Okay, don't do that. Wait, be patient. If you really want to take a probiotic, wait until they finally get regulated as a product. In fact, the FDA has even changed the name. They want to distance themselves so much from probiotics, they now call them live biotherapeutic products because they don't want to be associated with the probiotic industry. And you know, they sell, I mean, they sound amazing, don't they? But as long as they don't make a health claim, as long as they make some kind of squishy remark about it'll make you feel better or whatever, they can sell whatever they want to. But don't waste your money. And I will tell you, some of them can be dangerous. And I'm going to tell you an ugly little story. NICHD, National Children's Health and Development, mounted a big pediatric cohort. And one arm of the pediatric cohort study was to give these kids, these babies, a probiotic. The babies, some of the babies died. Okay, so probiotics aren't safe, they're not efficacious, they're not even regulated, they're a waste of your money, I swear, don't do it. Don't let your doctor say, oh yeah, I've given you an antibiotic, go out and get a probiotic. Don't do that. Yes, exactly, that's what she's reminding me. So instead, eat yogurt, eat fermented foods of all manner, okay, and eat fiber. Eat fiber and eat fermented foods, and I think, and be consistent. There's no miracles, but over time, you will benefit.
And sour poi.
What is it?
And sour poi.
And sour poi, pardon me, sure, of course. Okay, I don't know, what do we?
Out of time, but if you want to make a final remark to send us out into the microbiomic night.
Feed your microbiome and live a long and prosperous life.